Good morning, everyone, and good evening, or maybe good afternoon. Welcome to our today's System Thinking Series webinar. I'm Lian Teng. I'm your host today. I'm the editorial development manager for ASM Journal in China. And before we delve into today's webinar, I would like to give you a quick overview of our journals and systems. Can you please next? Okay, so mSystems encompasses all important biological and biochemical findings in microbial systems biology, including those drawn from analysis of large data sets, as well as new technical and computational methodologies for deriving these insights. And there are a few features that make mSystems stand out. The first is global reach. mSystem is a fully open access journals, which will give your research wide reach. And then on impact levels, mSystems welcomes data sets to support open science and reproducibilities. And we also ensure get the research published quickly with rigorous but quick peer review. Next, please. Thank you. So there are several mSystems initiatives is ongoing. The first is special series on social equity and disparities in microbial exposure. And those articles highlight the beneficial and the detrimental instances of microbial exposure in the context of how social policy may mediate or deepen disparities between and within populations. And another series is a companion series with microbiology resource announcements, which is also a ASMOA journals. This journal allows authors of accepted original research articles in mSystems to submit MRA companion articles, focus on expanded rigorous analysis of data resources reported in the primary article. Next, please. Okay, thank you for letting me introduce our journals, and let's get into our webinar today. Firstly, it's our great honor to have three brilliant researchers who are also mSystems editors join us as moderator and speakers today. As a starter, I will quickly introduce our moderator today, Dr. Jian Xu. So Dr. Shi is a professor and director of Single Cell Center and the director of Bioenergy Division at Qingdao Institute of Bioenergy and Bioprocess Technology, Chinese Academy of Science. He obtained BS in biotechnology from Peking University and MS in computer science and Ph.D. in biochemistry from Washington University in St. Louis in 2003. He joined Quebec in 2008, and his group focused on single-cell metabolic phenomenics and the rhamnomics instruments to support single-cell metabolic phenomenics and genomics profiling for industry, biotechnology, precision medicine, environmental remediation, and synthetic biology. His contribution was recognized by many awards from NSFC, MOST, and CAS. And Dr. Xu also serves as the senior editor of mSystems. So, and before I hand over to Dr. Xu, I would like to quickly introduce my editorial colleagues. They stay up late for today's webinar. And we have mSystems journal development journal development editor, Lauren Clark, and Noelle Lin, our managing editor, join us today. So if you have any question about this journal, they should be able to answer your question at the Q&A session. Okay, without further ado, Dr. Xu, please.
Thank you, Lian, for the nice introduction. And thanks to the mSystems to provide such a nice platform to discuss science and to exchange ideas with experts. So today, it is such an honor to have two extraordinary scientists as well as clinicians to give us talks on their expertise area. So let me introduce our first speaker today, Dr. Huang Shi. Professor Huang Shi is currently an assistant professor in microbiology at the Faculty of Dentistry. Yes, we are going to talk about mouth microbiome today as well. And then from University of Hong Kong, he's a computational biologist with a mixed specialty in microbiology and computer science. Over his years in training in China and also in United States, and now in Hong Kong University, he has developed many important computational tools. So let's welcome him today to give us a talk titled Methods Development for Analyzing Low Biomass Microbiomes in Human Clinical Samples. Welcome, Huang Shi. Please go ahead.
Can you see my screen now?
Yes, perfect.
Great. Thank you so much for a nice introduction. So today I'm going to be very happy to share with you my recent work on the method development for analyzing low biomass microbiomes in human clinical samples. Okay. So as you might know, we are not alone. We have only 50% of human cells and only 1% of the genes in our body. Our body is also a home to microbial cells, including bacteria, fungi, archaea, and virus, collectively known as human microbiome. It's profoundly associated with many host phenotypes, also revolutionized the method development for clinical diagnosis and treatment. So in the past decades, I worked together with our team, like building up a series of microbial index for predictions of human health, including microbial index of halitosis, periodontal health, dental caries, and also gut health, and then skin health. So two big examples is that number one is ECC demonstration. So ECC is actually one of the most prevalent dental problems inflicting 50% of children worldwide. Here we try to identify a suboptimal health stage of dental caries where oral microbiota. Here we demonstrated the tipping point of oral microbiome change or along the development of dental caries. And then we evaluate the capability of oral microbiota for predicting the onset of dental caries with the accuracy of 80% accuracy using machine learning technology. And then we also focus on another major dental problem worldwide, which is periodontal health. Here we establish a longitudinal multi-omics analysis and then microbiome meta-analysis technologies to link periodontal health with host aging. Interestingly, we found that as early as 3 days after pulse of dental hygiene practice, the molecular level signal of microbiome and metabolites and also host cytokines can be very similar to that has been observed in the very advanced periodontitis and elderly individuals. So in the past work, we pretty focused on the microbiome on particular body habitats, but we know like microbes would just stay at the surface of the body habitats. It can probably invade the tissue and then participate into the onset and progression of the oral chronic diseases. So that's why it is very critical to characterize the microbiome cohabitating with human cells. But it remains challenging. So all the problem can be classified into a big thing, which is a low biomass microbiome analysis issue. So these issues can be also categorized into 3 parts. Number one is HOC, which is called host contamination. For the representative sample type will be blood, saliva, tissue samples. It will usually contain 95 to 99% of human DNA in the resulting metagenomic analysis. And OA, which typically found in skin microbiome analysis. The big challenge is very low amount of DNA can be sequenced. And for FFPE sample, we call like this issue as HID, high degradation issue. So only less than 150% of BP DNA can be sequenced, which is not allowed for most like conventional approaches as a sitting S. So all these issues are very challenging for conventional approaches, namely sitting S, amplicon sequencing, and shotgun management sequencing. So now we are wondering if any alternative sequencing technology to identify microbes from a high host community. So for 16S and 18S and IDS amplicon sequencing, usually we will amplify a marker gene from each microbe to represent the percent of the microbes in the community. And it will lead to like a very low resolution taxonomy profiling results and by PCR bias taxonomy results, and also high off-target amplification issue, as we mentioned before. For WMS, we assume that we can sequence the whole full sequence of the each genome in the microbial community, but in reality, this is not going to happen because usually like the full sequence will not be fully sequenced in the management of the technology in the current stage. So now we wonder like if we can identify any like genome-wide markers like widely distributed along the genome that can be sequenced by certain ways that can pull out from the genome for sequencing. This can reduce the overall genome size and then can increase the overall coverage of each microbes in the community sequencing. So this type 2B resistance enzyme associated DNA is the one that we are choosing. So this can be also called a reduced genome-wide genotyping methods. It's been well established in periodic studies. It can produce like isolate fragments. It represents as a high reproducibility for quantitative measurement and low sensitivity to DNA degradation. So this is the typical chemical reactions can be found for a resistance enzyme like a CJP1 cut a genomes into thousands of millions of 28 BP long DNA fragments. So furthermore, with this rationale, we established a 2B-Rad M workflow, which contains two parts. Number one, sequencing part and bioinformatic part. In sequencing part, we do regular procedures as the metagenome sequencing. We extract DNA, and then typically we digest the DNA sample with type 2B resistance enzyme, and then add the adapters for amplification for DNA sequencing. And simultaneously, we establish a 2B-Rad marker database. We map the DNA sequencing reads to the DNA species specific 2B-Rad marker database for microbiome identification. And then we perform taxonomy profiling or abundance estimation using the sequencing coverages of each of a marker species-specific markers in the database for abundance estimation. So given this platform has been established, we're trying to see if this is accurate using the MOL community. More than that, we actually create a set of MOL community in simulating the host HOC condition, high host condition. So this is the MSA 1022 containing 20 bacteria. It's a DNA MOL community. We mix together with 90% of human DNA and 99% of human DNA. And for these two MOL community, we perform like 16S and then 2B-Rad and sequencing for taxon profiling at the genus level. And AUPR here represents the accuracy of microbiome identification, and L2 similarity represents the abundance estimation's accuracy. So as you can see, like overall 2B-Rad, the M outperforms 16S in the overall taxon profiling, especially 16S can result in huge numbers of false positive. And if you have large proportions of DNA from human mixing to the sample, the false positive can be even higher. So for species level profiling, the cysteine is not going to be work very well. But in the recent science paper, they introduced a 5R cysteine RNA sequencing method we introduced here as well for species level profiling. It seems like better than the V4, V5 cysteine analysis, but it still can introduce a huge number of false positives, white blocks here. And also, if you can see still like a large number of false positive after introducing more DNA, human DNA here, and biased, E. coli abundant estimations being observed in this for this method. But overall, 2B-RAD-M is certainly outperform all the other methods in species level profiling and stringency level profiling. And then we move this benchmarking work to real samples. First case is the like a high biomass fecal samples. We only collect 3 fecal samples with ultra deep DNA sequencing with WMS and 2B-RAD-M and also cysteine S. Overall, we are estimating the accuracies of this measure in like estimate abundance of the individual bacteria. So at the genus level, it shows a very high consistency with the cysteine S, and then at the species level, we also observe very high correlations between these two methods. Like you can reach up to like 97.4%. Okay, and then given we perform like ultra deep DNA sequencing, we know we can estimate the lowest sequencing depth required for accurate taxonomy profiling. As you might see here, only like 3 million reads per sample for is required for to be read in a species resolved consistent precise DNA the diversity estimation and taxonomy profiling. However, for the shotgun management, it requires at least 50 million reads per sample. And then we also tried this method in skin microbiome, which represents a very low biomass sample type. First, WMS failed to analyze the microbial constitution here, and then 2B-RAD-M can provide like either high accurate, highly correlated taxonomy profiling at the genus level and then species level. And then, remarkably, this method can simultaneously analyze the fungal composition in the, for example, car surface sample, home surface sample, and also underarm sample. So all the fungi found in these samples are biologically meaningful, as we know. And the most important thing is the saliva microbiome of our interest in the our faculty of dentistry. But this is actually very challenging sample type for microbiome sequencing, because only 50% of the human cell in the samples can result in significant increase of the host DNA in the, for example, in the resulting metagenome sequencing. For example, it can be 98% of the human DNA. So we initiate another saliva collection and microbiome study here. We collect the saliva from 8 adult individuals at 4 time points a day. And then for each sample, we perform metagenome analysis with cysteine S sequencing, shotgun metagenome sequencing, 2B-RAD sequencing. And overall, we observe very high consistency between the 2B-RAD-M and WMS. However, the cysteine S has a very relatively low consistency with WMS and 2B-RAD-M. It means cysteine S is not a good way to analyze that is, genus level composition as well. Sorry. At the species level, we observe very high consistency between 2B-RAD-M and WMS. So this indicated high taxonomy composition consistency can be between like 2B-RAD-M and the WMS. And then we next perform the cross-platform comparison of beta diversity analysis. So here at the genus level, each DOS means the beta diversity measured by 2B-RAD-M or WMS. So you can see very high consistency between two masters. However, mSystems and WMS at the genus level only reach to less than 50% of correlation in terms of beta diversity measurement. Here is the recurrence. At the species level, we observe even higher correlations between beta diversity between two masters in terms of beta recurrence and weighted unit frag. Like mSystems perform even fully in is estimating beta diversity. So now we can move to, we are now convinced ourselves to move to even challenging microbiome present in the FFPE sample, which is the most rich resource in the King hospital. Okay, we here collect 45 cervical FFPE samples from healthy precancer, invasive cancer for alpha diversity analysis and beta diversity analysis. Here we resulting a very reasonable, biologically reasonable results where we observe a very low diversity and dominated by lactobacillus microbiome in the healthy cervical samples, while all the cancers has very high diversity and then enriched by other different kinds of pathogenic bacteria. And machine learning model based on the fecal FFPE microbiome resulting a model predicting cancer states with accuracy of 91% accuracy. So recently, we also got the funding from HMF from Hong Kong to discover the microbial features of oral cancer diagnosis and prediction. Here we have a established Hong Kong cohort. We can collect FFPE samples from two group of people, which is OPMD to OC, OPMD to OPMD. OPMD is a precancer status of oral cancer. OC is oral cancer. Okay, so OPM, we are trying to establish a machine learning model using FFPE microbiome analyzed by 2B-RAD-M to establish a prediction model for OC at the stages of OPMD, and then predict the OC probability for OPMD to a OPMD patient, and then predict the future outcome of these patients in the following visits. And interestingly, oral tissue microbiome tends to be more diverse in oral cancer as compared to OPMD. This is the alpha diversity, a difference of alpha diversity, also beta diversity measurement, also support this kind of idea. And then if you take a look at into the heat map here, you can certainly tell which one is like OC status. This is one's OC status. Much more microbes can be detected in the OC status as compared to OPMD status. Okay, but we think that these markers can be very promising markers for predicting OC from the status of OPMD. And then we are not only stay on like at the species level providing using 2B-RAD-M, but we are well aware of the widespread amount of strain level analysis of microbiome, especially in the low biomass condition. For example, conventional metagenomics method requires high sequencing coverage and limited ability to identify strains in low biomass samples. So we are aiming to address these questions with challenges with our technology. So first, we try to see if 2B-RAD sequence extract from, for example, existing full genome from database can distinguish strains having identical sequence, 16S sequence. So here are 27 genomes from Cutibacteria acnes, and this is the 16S phylogeny tree, and this is the 2B-RAD phylogeny tree, and this is the whole genome phylogeny tree. As a gold standard, whole genome data being tested very similar to what we established based on 2B-RAD-M, but for 16S, many strains cannot be distinguished And this is more quantitative analysis of the last phylogeny tree. So where we put the distance from each pair of genome together using 2B read and whole genome and 16S. And you can see a perfect correlations observed for 2B read and whole genome distance. But 16S usually will have a very low correlation. You can see here, like for example, for those identical sequence genome with identical 16S sequence, they can have a wide ranges of distance in the whole genome. Okay. And then here is the rationale for strain level profiling based on 2B read sequence. First, you can establish a copy number matrix using the select genome from the database, and then you can get the reads count from the sequencing platform. Okay, so this was simple 1, simple 2, simple n. And then you want to estimate species abundance. And this equation actually tell you how we calculate this metric computation, how you calculate the strain level abundance. Okay, here is the constraint ordinate least square algorithms that have been used for analyzing this. And this is a strain level accuracy testing. We know this is very accurate with simulation metagenome. And then we further move forward to functional profiling. And this is the basic the rationale, and then this is the resulting benchmarking method showing the accurate functional estimation with our method. Anyway, this is a big summary. Like we are having this method not only for strain level, species level, but also strain level analysis. But we are also moving forward to benchmark this method on many more real clinical samples. In the future, we are more interested in moving forward from the 2B read based metagenome analysis to a functional driven single cell omics and culture method. This method can be driven by our recently established platform, which is called Raman technique platform, incorporating single cell Raman microscopy measurement, sorting, and sequencing culture omics. Basically, we can observe capture the metabolic phenome from single cell level, and then do functional sorting based on this signal, and then perform single cell level genome sequencing and transcriptome sequencing, and also cultivation. So this is the basic Raman concept. We can collect each Raman spectrum for each single cell at a single cell and a cellular population, and then based on the fingerprint region to tell which bacteria is which. And then for at the silent region, we can tell if this bacteria metabolic active to under certain conditions. So here we show many applications of these technologies, for example, anabolism, catabolism, phylogenetic distinguishing, species activation and interaction, stress response testing, and also metabolic network. So this is our main goal for HKU platform, where I'm a PI. So including antibiotic resistant tests, and then single cell functional sorting, mutant library screening, and human cell fate prediction, and also human cells sorting. So let's go back to the microbiome analysis of host-rich samples. For this goal, we are proposing to utilize this very unique platform with our existing metagenomic capability to analyze the tumor liquid biopsy in a high throughput manner, and also test the tumor drug resistance with flow rags, for example, one of machines in our core facility, and also intra-tumor tissue microbiome with spatial Raman and flow rags, and also single cell sequencing, single cell cultivation technology. So anyway, we are very optimistic on our future on analyzing the microbiome with two strong capabilities. Number one is the 2B read driven metagenomics analysis, also Raman activate single cell competency driven single cell level analysis of microbiome in high host samples, clinical sample. I welcome to every clinicians and other collaborators from anywhere to work together with us to enhance this capability and then to show many interesting biological story to the world scientist community. Thank you so much. This is acknowledgement to all our collaborators and then our funding supports. Thank you so much.
Thank you, Huang Shi. Thank you for a very exciting talk. Because we are running out of time, so let's leave questions to the later Q&A session. Okay. Thank you, Shi, for the nice talk.
Sharing. Thank you.
Thank you. Now let's move to our next speaker. This is an extraordinary scientist and clinician. Professor Zhou Hongwei is Director of Microbiome Medical Medicine Center and Director of Division of Laboratory Medicine of Zhujiang Hospital at Southern Medical University at Guangzhou, China. He has pioneered in linking the gut microbiome features with regionality, and truly one of the pioneers in this field. Let's welcome Professor. Let's take this moment to answer a few questions from the audience. Then first, Professor Huang Shi. Hello, can you hear me?
Yes, sure.
Yes, we have actually 3 questions. Very nice. First question is, was skin the source of fungal samples in case 2? What was the source of fungal analysis? Can you clarify?
We are not pretty sure what is the source. I mean, this fungi was detected on from the sample underarm. I assume there will certainly be some fungal there, as we talked before.
Armpit, right? So
yes.
the underarm. Okay. We saw Professor.
So thank you very much, and I'm sorry for this. Today we are going to talk about the clinical practice, preclinical microbiome. So before we can apply this understanding of the microbiome field into the clinical practice, we try to understand the causative role of human microbiome in chronic diseases. Just like Shi just talked that the human microbiome is correlated with a wide variety of chronic diseases, from the pregnancy to the aging progresses. And so for all of these chronic diseases, we want to answer a specific question, which is what keystone strain or keystone function play a critical role in the specific disease, and what are the molecular mechanisms underlying these progresses. So I will introduce you our understanding of this specific progress with the 4 recent findings from our lab, which are specifically the preeclampsia, ischemia stroke, Crohn's disease, and the rheumatoid arthritis. All of these diseases are traditionally the chronic diseases. Let's start from the preeclampsia. So as we know that the preeclampsia is a sort of hypertension during the gestation after 20 weeks of the gestation, and the pathology of this disease is quite obscure until now. So we start from the collection of the fecal sample from the preeclampsia patient and the normal tension pregnancy woman, and we perform the 16S RNA sequencing, and we can see that according to the PCOA analysis, these 2 groups can be clearly separated from each other, and also the alpha diversity also show differences between these 2 groups. And with the correlation analysis, we can see that the genus of the Fusobacterium and the Veillonella are positively correlated with the blood pressure and the proteinuria, while the Akkermansia and the Faecalibacterium are negatively correlated, which indicate that the gut microbiome may play a role in the preeclampsia. So furthermore, we inoculated the fecal material from the pregnancy woman to the mice, to the antibiotic treated mice, and we see that after the fecal material transportation of the gut microbiome from the pregnancy woman, we can see from this PE patient group, the blood pressure of this animal model group significantly increased, while in the normal tension group, the blood pressure still keeps normal. And before the gastration, we can see that before the delivery, we can see this urine protein also significantly increased in the PE FMT group, and also we see the higher ratio of fetal resorption in this PE FMT group as well. So all of these phenotypes are pretty much similar to the preeclampsia patients. So indicating that there are a causative rule between the gut microbiota with the preeclampsia. And furthermore, we can see that from both the qPCR and the FISH analysis, we use the placenta samples. And on the placenta samples of the normal tension patient and the preeclampsia patient, we can see from these preeclampsia samples, the signals of the bacteria significantly increased in the PE group, and also in the animal model after the fecal material transplantation, we can see from this PE group the bacteria signal also increased from both the 16S-RNA qPCR and also the FISH analysis, and also we can see the increased inflammation in both the patient and the animals as well. All of these indicating that there's potentially a causative rule between these two, between the gut microbiome with the preeclampsia. So after we published this paper at that 2020, Dr. Hampton wrote a comment in the JAMA asking that, do gut bacteria play a role in preeclampsia? So after that year, we performed a further study, and we know that for a specific bacteria after we inoculate a specific bacteria into mice, it can induce all the phenotypes which is similar to the preeclampsia on the animal model. So, and more than that, we are performing a CALM 2000 maternal cohort study. In this study, we have longitudinal cohort, one center for continuous samples, and 3 to 5 samples centers for one-time sampling during early pregnancy. We want to see if the gut microbiota and also their metabolites during the early pregnancy can predict the outcome, can predict the occurrence of the preeclampsia. And also we have the cross-sectional cohort as well. Next is the ischemia stroke. And we know that all of these chronic diseases are correlated with the dysbiosis, and they are the basic diseases for the stroke. But for this specific study, we are working on the acute stage of stroke. We found that there is a rapid gut dysbiosis induced by stroke, and then exacerbate the brain fracture in turn, which is a vicious circle from the brain to gut, and then from the gut to brain. For this specific progress, the road is that the stroke can induce the ischemia reperfusion of the gut through the excitement of the vagus nerve, and we can see all of this ischemia reperfusion genes significantly increased their expression, and they produce a large amount of free radicals in the gut lumen. All of these are excreted in the gut lumen and finally produce nitrate oxide. And we know that in the gut lumen, it is an anoxide environment with bacteria. This progress, I mean that from the brain to the gut, this progress with the brain ischemia induced the intestinal ischemia reperfusion and the overgrowth of the enterobacteria through nitrate respiration. And then we can see from the patient in the gut lumen. And after this overgrowth of the gut enterobacteria, and it can impair the brain function through the LPS-TL4 pathway. We can see that after we inoculate the LPS, the E. coli into the antibiotic-treated mice, the brain, after that, we do the MCL modeling, we can see the brain function significantly increased. While we knock out the LPS gene, we can see the brain function goes back to the normal level. And we know, to further support this hypothesis, we used the aminoguanidine to reduce the production of nitric oxide, and also the SOD to remove the superoxide, and also the transtate to reduce the nitrate respiration. All of these inhibitors, we can see that they significantly decreased the level of Enterobacter CA in the gut, and also the brain function also significant, not only significant, but very obviously decreased after with these three sort of inhibitors. And interestingly, after we use the antibiotics to remove the gut bacteria, we can see that these three inhibitors cannot work after this process. So all of this indicated that these inhibitors are dependent on the gut microbiome to work during this circle of the from the brain to gut and then from the gut to brain. So after this study, we performed a COM200 study we use, which is focusing on the ischemia stroke cohort, and we recruit during the admission stage, and then after the discharge, we collect samples from the 6 to 12 months, and we want to ask if the gut microbiota can predict the prognosis or the relapse of the stroke, and also the depressions and the PSCI as well. So the third one is Crohn's disease. As we know that for the Crohn's disease, the traditional mechanism is the NOT2 mutation. However, as we know that even in the Western countries, the mutation of the NOT2 takes about 20% of the patient, and in China, the percentage of the NOT2 mutation in Crohn's disease patients is pretty low in China. So in this study, we make assumptions that for those patients, they have intact NOT2. However, because the NOT2, the ligand is MDP, which is shed from the cell wall of the gut microbiota. So maybe which is because of the gut microbiome dysbiosis, which induce the NOT2 ligand is not sufficient. Therefore, the activation of NOT2 is not sufficient, which induce this Crohn's disease progress. So with this assumption, we analyzed the genome of this more than 1,500 strands from the gut bacteria, and we evaluated the 8 types of peptide glycan hydrolysases in these genomes. We find that this DL endopeptidase could be a limitations enzyme because it only exists in very limited number of genomes. And furthermore, we analyzed more than 2,000 metagenome samples, and we find that this DL endopeptidase is pretty low, which further support that it could be a limitation enzyme. And then we analyzed the 4 Crohn's disease cohorts from IHMP, a Spain cohort, a China cohort, and Netherlands cohort. For all of these 4 cohorts, we can see that the level of DL endopeptide genes are significantly negatively correlated with the colitis activity, which support there could be a correlative rule between these two. And then we do the FMT study on the animal model, and you can see that the lower of the DL endopeptidase, the more severe of the phenotype of the model, which indicated that there could be there should be a correlative rule between this DL endopeptidase level with this Crohn's disease. And after that, we use both either from the DL endopeptidase, the recombinant endopeptidase, or the specific probiotics encoding this enzyme, or the MDP analogs. All of these drugs can significantly ameliorate the severity of the disease. So after the publication of this story, the Cell Host write a comment that this ligand could become an antidote of this Crohn's disease. So the last one, I will talk about the rheumatoid arthritis. As we know that the arthritis, RA is a very complicated disease, and we know that it have a cyclic cycle as well. So we collect samples from the fecal samples from patients during the morning time and the evening time, and we also compare that with RA patients with control patients as well. We find that their gut microbiota have very significant difference between this time set and also from the healthy to the RA patient as well. So from this analysis, we focused on the Fusobacterium nucleatum. For this specific bacteria, after we inoculated this bacteria into the mice and we do the CIA modeling, we can see that comparing to the E. coli group, the Fusobacterium nucleatum group significantly increased the inflammation of this mice model. And for this specific bacteria, we tested many approaches, and finally we find that it induced the RA inflammation through the outer membrane vesicle, which is because when we used this inhibitor of the OMV, the inflammation significantly decreased. And after we extract the OMVs out and we inoculate this OMVs into the mice model, we can see that it can accumulate in this joint section, in the joint positions. And with this two different OMVs from the Fusobacterium nucleatum or from E. coli, we can see the OMV from FN can significantly increase the inflammation as well. So for this specific OMV, we further find that the fat A protein of this OMV is very crucial, because while we knock out this fat A protein, that OMV cannot increase the inflammation level at the animal model, in the animal model. So very interestingly, that we can see that the Fusobacterium nucleatum derived fat A is determined in the OMV from the synovial fluid of the RA patient, while in the control group we cannot find any sort of this fat A protein, because this fat A is encoded in the Fusobacterium nucleatum. And also from the synovium of this patient, we can see that from the fat A to RAC5A and to YB1, all of these are more abundant in the patient group. So this from this study, we propose a new pathway of this specific RA disease of this pathology from the gut bacterium from the Fusobacterium nucleatum to the OMV, and then it can translocate to the joint position, and then induce this inflammation progresses. So from this, according to all of these different types of studies, we developed this Chinese Association for Clinical Microbiome, which is CAM, and we performed a lot of cohort studies, multi-center cohort studies from the maternal ischemia, genital microbiome, and preterm chronic and IBD as well. So for all of this is what we are performing the multi-center studies. So we want to, because we know that the gut microbiome is very significantly different between different locations. We need to have this sort of multi-center studies according to the different regional dependent. So for now, we have the microbiome, correlative microbiome study. We found the correlation to causation, and then finally to the translation. For all of these chronic diseases, may we ask a question: is this a new era of the pathogen? Because which is very different to the original era of the pathogen, which is focused on the infectious disease. But right now, we can maybe transfer to the chronic diseases from this new stage of the new era of the microbiome. So in our lab, we have interest in the gut-brain axis, gut-liver axis, and the maternal and newborn microbiome. So with this and thank you very much for that's all.
Hi, thanks so much, Professor Zhou Hongwei. You had a spectacular, really, talk. The new hypothesis for the new versions of the causality in microbiome-based disease, I think it's truly a new area to explore. And so we actually have quite a few questions for you. Let me start with the first. What type of samples will be collected in the PE study at the time of birth? How did you determine the best or variety of sample to collect? Are they all placenta samples?
Oh, okay. We collect 3 types of samples from at the time of birth. So we collect the placenta samples, we collect fecal samples, we also collect blood samples as well. For the placenta samples, we do not collect all placenta, but a very specific site of the placenta. We have a protocol for that.
Excellent. So another question here, which is also very thoughtful, is that have you looked at the Veillonella species variants? Recent research has suggested a role in creating biofilms with hospital environment for anaerobes. This could be related to your Veillonella and Fusobacterium correlation. Very interesting.
Yes, yes, this is a very interesting question. Actually, we have not yet looked at the Veillonella species right now because there are so many different species. Even in the Fusobacterium, right now we are focusing on it because there are many different species in the Fusobacterium. So we are still working on the Fusobacterium right now. But yeah, I think Veillonella is also very, very important to work on as well.
Maybe you can consider working with Huang Shi on his strain level resolution to be RAM. So you can look at many different strains of Fusobacterium, for example. Okay. So third question is that have you considered the biofilm on the interior joint? Have you?
Not yet, because we actually for that joint, for that rheumatoid arthritis, we actually did not find any bacteria in the joint fluid, but rather we find the extracellular vesicle, not the alive bacteria.
Excellent. Now another question is about a suggestion. We need to consider relationships of microbes with the microbiome. I guess it's intermicrobial communication. Do you think we should consider the microbial communications with biofilms? This is to Hongwei.
Okay, I think this is you are expert in this field. Actually, I'm not quite an expert in this biofilm field, but yes, I agree with you. I agree with with this question that the interaction of this of very specific bacteria is very important to understand. I think that maybe with the Ramanon technique, may be a useful technique to study biofilm. But I think that in the gut microbiome to chronic diseases, right now there are not so many studies on this topic.
Yes, Shi, do you have any comments along this question?
I think,
for example,
yes,
how do you use Raman to map intercellular communications? For example, exchange of metabolites, either between bacteria to bacteria or between bacteria and fungi or between bacteria and host cancer cells, for example.
Yes, and now is also proposing a series of studies to understand, for example, the interactions between H. pylori and oral bacteria using Raman microscopy platform. So we're trying to see if co-culture, for example, oral to gut or stomach microbiota location can increase the antibiotic resistance of H. pylori or vice versa. Because recently, CUHK's Professor Yu Jun has published a paper on the translocations of oral-derived strain Streptococcus aeruginosus into the stomach and lead to gastric cancer. So there are much interactions between oral bacteria and gastric bacteria not being uncovered. Yeah.
Excellent. So I guess the bottom line is that by developing new tools for both community level and single cell level, both function-based and sequencing-based, basically genotype-based approaches should help us to really get down to the mechanism level about such inter-host fungal cell and bacterial cell communications. Okay, the last question is how gut influence the liver? I guess this is for Hongwei. This is a very broad question, but
yes, a lot of approaches spanning from the Ph.D. to liver cancer. I think that a lot of metabolites from the gut can influence the liver function. There are many papers.
This literature reservoir about this topic. This is very important new topic. How gut microbiome affects liver? Yes.
Yeah, I agree.
Okay, we probably don't have time to go into depth of this important question. But hey, we also have two senior staff members from ASM. Editor Noel Lin, he's the manager editor of the journal, as well as Lauren Clark, and who is also an administrator for this journal. So if there are any questions for our journal, I guess we are open to discussion. Hi, also Noel, Lorraine, would you like to say anything?
I just want to thank everyone for joining today and also for the speakers, really excellent talks. And also thank you, Jian, for managing the questions, and to Leon for organizing and leading the introduction.
Thank you, thank you. I guess Asia and America, they are the two biggest hubs of microbiologists in the globe. Hopefully we can, turn this in such thinking series into routine, so that, microbiology research, they doesn't stop, right? They do around the clock. Okay, so Leanne, do you have any final words to say to the audience and to the speakers?
No, yeah, I just want to say thank you for all for Dr. Xu and for Dr. Zhou and Dr. Huang and Lauren and Noel for joining us today, and thank you for all the attendees and all the great questions. And I hope you have seen the email address from the speakers and the moderator. So if you have any further question, either no matter both from either from for the scientific side or for the journal, please don't hesitate to reach out to us. Thank you for everything. Yeah.
Thank you. I just want to add that I almost forgot. Actually, there are quite a few very thoughtful questions for Huang Shi's talk as well. And Professor Huang has taken the effort to write very detailed responses on the screen to the audience. So I guess everyone can take a look. Okay, now we are at 60 minutes. Well, let's thank everyone again for attending this seminar. Thank you, Professor Zhou and Professor Huang. See you next time.
Yeah.